Hi, I'm Melika, and today me and my partner Alex will be presenting our project analysis of gravitropism in natural populations of the plant Arabidopsis thaliana. So during long-term space missions, astronauts depend on plants for food. And in this project, we're looking for plant varieties that will grow well in space. So normally plant shoots grow towards against the gravity vector, which is upward, and the roots grow towards the gravity vector. But in space, since there is little to no gravity called microgravity, plants can experience gravitational stress, which can negatively impact their growth. So we're looking for plant varieties that can have this stress resistance and can grow well in space. And here on the right, we can see an example of plants that are growing vertically, so normal conditions, and on the bottom, plants under gravitational stress, simulated gravitational stress. We're using Arabidopsis thaliana because it's a great model plant. It has its whole genome sequenced and has counterparts that are edible. So if we find genes that are responsible for gravitational stress resistance, those plants could be manipulated and make plants suitable for growing in space. So our objective was to test 150 strains of Arabidopsis thaliana for their gravitational stress resistance using this device on the left called a 2D clinostat that simulates microgravity by spinning and basically confusing the plant as to which way the gravity vector is. And so our methods were to surface sterilize 51 genotypes of Arabidopsis thaliana, and they were placed on a nutrient agar, and then after 7 days of treatment, which was either clinostat treatment or vertical treatment as a control, they were scanned and analyzed using a image analysis software, and several parameters were analyzed using the PG software, including the shoot length, the main root length, the total root length, and their number of root hairs. And to find the difference between the different groups, we used a t-test to create the graphs.
Under clinorotation, we found 49 genotypes showed reduced shoot growth, and 2 genotypes, CIBC-5 and Col-0, showed no significant difference from controls in growth. Under clinorotation, 7 genotypes showed an increase and 20 genotypes showed a reduction in root growth in comparison to the controls. CIBC-5 and Col-0 and 22 others showed no difference in root growth from their controls. Under clinorotation, 19 genotypes showed a reduction and 6 genotypes showed an increase in main root length in comparison to the controls. We found a variation in growth responses across the wild-type genotypes. Our data suggests that clinorotation leads to reduced shoot length in the majority of seedlings. 7 genotypes out of 51 showed increased total root length under clinorotation. 6 genotypes showed increased main root length under clinorotation. Two genotypes, Col-0 and CIBC-5, showed no difference or better growth in shoot and root length from the controls and may have a potential for gravitational stress resistance. With our results, we think that the genotypes CIBC-5 and Col-0 may be prime candidates for future space experiments with plants. Further studies will aim to identify genes involved in developing resistance to gravitational stress. In the long term, these studies will help us to select plants to support astronauts in longer space flight missions. Thank you for watching our presentation, and feel free to ask questions.